welcome to the part 2 of which is the light microscopy of the chapter 4. And in this video, we are going to only cover the part 2A, which will be the Brightfield microscopy. And then we will cover the part 2B as the fluorescence microscopy, which is also under the section 4.2. So let's take a look at the outline. 4.2a, light microscopy, exploring cell structure, visualizing the proteins within the cell. Light microscopy, couple of resolution, wavelength. We're going to talk about some of the approaches to increase the effective resolution, and we are going to talk about the refractive index and staining procedure as well. So let's start. So microscopy, the average dimension of a typical animal cell is about 10 to 30 microns in diameter, which is about 5 times smaller than the smallest particle actually visible to the naked eye. The first good microscope was developed in the 19th century. At that time, it was first discovered that the plant and the animal tissues are actually aggregates of individual cells, and the idea or discovery is known as the cell doctrine and makes the birth of cell biology. So the early microscopes, like the one used by Robert Hooke, used lenses or mirror to illuminate the specimen and first reveal the existence of cells, which revolutionized science. Now, development of optics and light microscopes during the 19th century led to the mid 20th century a highly sophisticated microscope, but limited only by a resolution of light. In the second half of the 20th century, the fluorescence microscopy, confocal techniques in the recent past, and several super resolution microscopic techniques pushed the light microscopic resolution into the nanometer scale, previously thought impossible without an electron microscope. But we are only going to talk about the Brightfield microscope in this section, and we are going to talk about the fluorescence and the other types of microscopy, which is also used light, but are more sophisticated, into the section 2B of this talk. So what is the most difficult aspect of observing the small cell if you ask a typical person? The most common answer is magnification, and is the major challenge for observing smaller structures. There are, however, some other challenges to consider. Another is resolution, and the third is a proper sample preparation. So we'll take a look at each of these challenges individually. So the magnification is actually the function the scientist uses, and in particular, it is the function of a specific lens. That is the objective lens. That is right here is the objective lens. Here in this figure is a setup of a simple microscope or a Brightfield microscope. The red line has been added to show the light path through the microscope. So there is a light source, a lamp, a series of collector lenses, a mirror, condenser to focus the light on the specimen. I want to mention here that the specimen for the light microscopes are usually mounted on a clear glass or plastic slide or a clear or glass plastic dishes, and they sit in a stage and usually adjustable right here. So once the light actually passes through the specimen, it is collected it's collected by the objective lens. And again, it is the objective lens which provides the most of the magnification in the light microscopy. In this example of the objective lens, 100x or 100-fold magnification is the objective, as you can see. Most single light microscopes have adjustable objectives, which can be usually 4x, 10x, 40x, sometimes 60x, and 100x. These type of microscopes or objective lens are probably the type that you are most familiar with. So the light passes through the objective lens and then often gives an additional magnification in this. It is a 10x, usually the most common one on most light microscope. So in this example, it is a 10x, which is the eyepiece. Usually the eyepieces or the projection lenses, which is right here, projection lens or the eyepiece that is called, sometimes it's called ocular. That's usually can be even 15x or 20x, but usually never more than 20x. But most common is a 10x. So the light passes through the objective lens and then through the projection lens. It often gives an additional magnification. And this example, it is 10x, which is usually the most common. So in this example, the final magnification is the multiplication of 100 with 10, and the total final magnification is 1000x. From the projection lens, the light passes to a detector, and today the detector can be a computer screen, but most often the detector is just simply your eye right here. That's the reason it's called an ocular or eyepiece, but more sophisticated microscopes actually do have a computer detector here, which is fed into a computer screen. So that was magnification. Now let's talk about the resolution. A very simple definition of resolution is the ability to distinguish between two objects. All microscopy depends on beams of radiation, such as light or electrons. And if you'll remember from your physics classes, the beams of radiation travel in waves. And the wavelength of a beam of radiation is denoted by the Greek letter lambda. The wavelength is the distance between the two valleys or two peaks, either two valleys or the two peaks of the wave. Now, resolution depends on the wavelength of a beam of radiation you are using. A beam cannot be used to probe structure smaller than the wavelength. Very important. Repeating, the resolution depends on the wavelength of a beam of radiation you are using. A beam cannot be used to probe structure smaller than the wavelength. So the light microscopy limits are set by the wavelengths of visible light, which is in the range of about 0.45 micron to about 7 micron deep red. So light microscopy cannot be used to probe structure with a distance of less than 0.45 micron between them. I will elaborate so that you can fully understand the concept. So the resolution is denoted by the letter D, and here is a second definition so you can understand the concept. The resolution is the minimum distance between the two distinguishable objects. For instance, if your microscope system has a resolution of 0.5 micron, it means you can never distinguish objects less than 0.5 microns or micrometer apart. If less than, the objects will appear as just one. So how do you calculate resolution? So here is the formula for resolution. The resolution is directly proportional to 0.61 lambda or wavelength, where the 0.61 is actually a constant in this formula, and the resolution is inversely proportional to the value of the numerical aperture, or n sine alpha. Here n is the refractive index of the media the specimen is sitting in under the microscope. All specimens are in some type of media. It could be air, it could be an immersion oil, or it could be even water. And all of the media have a refractive index, which is represented by the n in this formula. The alpha is the angular aperture of the microscope system that you are using. So you understand, let's take a look at the simple diagram of the Brightfield microscope so you can see the light is collected by a condenser lens and focused on the specimen, and the light passes through the specimen and collected by the objective lens and projected onto a detector. When the light hits the specimen, it hits at a particular angle right here, at a particular angle, and this angle is 2 alpha. This angle is 2 alpha, so alpha is half of the angle. So let's do a simple calculation. So what is the best resolution you can obtain from a simple Brightfield microscope? So the best maximum angular aperture you can get today is 70 degrees. As previously mentioned, the shortest wavelength of light is about 0.4 micron, and the best medium of the immersion oil is refractive index about 1.5. So if you put all these values into the formula, the result is 0.194 micron, or approximately about 0.2 micrometer or micron. This will appear as, so this is the resolution of a light microscope. So anything smaller than 0.2 microns will appear as one object when conducting simple light microscopy. So here is the image of a Brightfield microscopy. The image is good. You can see the image of the cell and some inner detail, but you may make out some of the organelles as well, but really it is not that great. So there are some advantages and disadvantages of simple Brightfield microscopy. The advantages are simple, inexpensive microscopes are readily available and not cost prohibitive. You do not have to stain the sample to see some kind of image, and you can image the live cells because there is minimum manipulation of the samples, so you can keep the cells alive. But there are some disadvantages or limitations. There is low contrast. Most of the biological specimen actually are composed of primarily of water, and as discussed previously, water which makes it for a low contrast of the specimen, and it is limited to the resolution of 0.2 microns. So let me repeat that. But there are some disadvantages or limitations. There is low contrast of most biological samples because most biological samples are composed of primarily of water, which makes for low contrast of the specimen. As discussed previously, there is low resolution, 0.2 microns at the best can be achieved, which is good, but you can do better. Another problem with the light microscopy is that you are collecting the light from the plane of focus and collecting the spurious light from the outside the plane of focus, which blurs the resolution or make the resolution worse. But there are some optical, physical, and biochemical ways to minimize the limitations. So let's take a look at some of the optical methods to improve the light microscope. One of the ways is to use the different types of light microscope. Like several different types have been invented, and it's the physics of the light microscope that make them differ from the standard light microscope. Now, I will not go into too much of detail, just touching some of the bases of the various scopes, but I will mention a few differences as well. In the Brightfield light microscopy, light from a tungsten lamp is actually focused onto the specimen, usually stained with dyes to enhance contrast by a condenser lens below the stage. On the other hand, in the phase contrast microscopy, which increases the contrast of the biological specimens, incident light passing through an annular diaphragm focuses a circular annulus or ring of light on the specimen. The light passing unobstructed through the specimen is focused by the objective lens into a thicker gray area right here, which absorbs some of the direct light and alters its phase by just 1/4 of a wavelength. If a specimen actually refracts or bends or diffracts the light because of the refractive index right here, refractive index of the material, the phase of some light waves is altered, which becomes right here in the middle part. These are the green lines. And the light wave passed through the clear region of the faceplate, and the refracted and unrefracted light is then recombined by the image plane to form the image. So that's how a phase contrast microscope actually works. So the differential contrast microscope contains the polarizing element typically encountered on a polarized light microscope, and then in addition, two specially constructed birefringent compound prisms. These are known as Wollaston or Nomarski prism. These optical beam splitter and beam combiners, beam splitter and beam combiners are positioned to project the interference pattern of the sheared wavefronts into the condenser in the front focal plane and to the objective near the rear focal plane. So what happens in this process, the semi-coherent wavefront, which is formed the localized neighborhood in the lamp filament, pass through a linear polarizer positioned between the light and in the microscope base and the condenser assembly. A compound Nomarski prism is located near the condenser aperture, which is the front focal plane, and serves to align and shear the incident polarized wavefront into these two orthogonal components. The perpendicular sheared wavefronts are focused by the condenser lens into the parallel bundles that actually traverse the specimen plane and respond to the refractive index and thickness gradient by deformation according to the optical path length parameter of the specimen. So depending upon the specimen's shape, size, refractive index, these two orthogonal components will vary in their phases. And this is the basis of this Nomarski interference. So the light wave gathered by the objective converge at near the focal plane, where a second Nomarski prism recombines the sheared and the deformed wavefronts into a linear and elliptically polarized light, and subsequently that passes through the component through an analyzer. And this is actually a second polarizer oriented perpendicular to the first one. And finally, that linear polarized light actually emerge from the analyzer, and the recombination through the both actually through the constructive and the destructive interference forms the image at the eyepiece. And in reality, the phase contrast and differential interference microscope or Brightfield microscope, they can all be built into a single microscope itself. So you can see that this is an universal condenser turret, which is attached into the middle, and you can just rotate these under the condenser to have Brightfield, Nomarski interference, or the phase contrast microscopy at your discretion. So let's compare the images from these microscopes. The first variation of the standard microscope is the phase contrast light microscope. As mentioned previously, the medium that the sample is in has a typical refractive index, but all of the different components of the cell have different refractive index depending on their makeup. So the nucleus has a different refractive index, the vesicles have different refractive index, Golgi apparatus, mitochondria have different refractive index. So the phase contrast microscopy takes advantage of these differences. When light is projected onto the sample, a series of annular rings, depending on the refractive index of the inner cellular components, it will push light out of phase or keep light in phase. Light out of phase is dark, and light in phase appears very light. So the result you can see is in more detail. See the comparison between the Brightfield versus the phase contrast images. You can appreciate the detail just gained by the phase contrast microscopy compared to the Brightfield microscopy, and capitalizes the different refractive indices of cellular components and gives a greater detail of the organelles. In contrast, you can see that the differential interference contrast, or DIC, or Nomarski interference microscopy, relies on the polarized light, produces a very different image but very useful. With DIC, you can appreciate that the image is almost a 3D image or a pseudo image of the cell. So you can gain some information about the three-dimensional structure or characteristic of the cell. So these are some of the variations of the light microscopy. So there are some optical methods to improve light microscopy, and what are some physical and biochemical methods? So now we need to look at the third challenge. How do you handle and prepare your samples before microscopy? Good sample preparation can actually improve or simple light microscopy. Sample preparation occurs in 4 steps. The first is a fixation step. Second is embedding. Third is a sectioning, and fourth is a staining step. The embedding and the sectioning steps are for thick specimens only, like tissue samples. Cells growing in culture don't require these 2 steps and can be directly fixed and then stained. And let's take a look at them individually. So start with fixation. So the cells are in dynamic state and the organelles are moving and the vesicles are being trafficked. Motile cells such as macrophage, neutrophil are on the go. lower eukaryotic cells are motile. All the movement complicates microscopy because as the cells are moving around, the resolution is lost. So the image is blurred. So fixation takes care of actually freezing the cells and stops all movement. It also kills them, and which may be good or bad. Fixation freezes the cells in time and stop all the motion so you can capture a good image. The way it freezes is by cross-linking macromolecules. It is not like a temperature freezing, it is a freezing in time by a chemical method. The agents used for the cross-linking are usually glutaraldehyde or formaldehyde, which actually form covalent bonds between the free amino group of the proteins, and fixation also can act to partially permeabilizing the specimen. So the second step in the sample preparation is sectioning and applies only to the sick specimen. If you are looking at a single layer of cells, then the sectioning is not required. But if you are looking at a tissue specimen from plants and animals, some sectioning is necessary. Some biological samples are too thick and light can't pass through them. You have to have a slice, thin section, so light can pass through, so you can capture an image on the detector. Most biological tissue samples are too soft or flimsy and sections, so you have to fortify or embed them. To do this, first you put your samples into some kind of liquid embedding material, such as molten wax. Some commonly now put or paraffin, and sample onto a liquid polymer or plastic. And after either cooling of a wax or paraffin or polymerizing of the plastic, you end up with a solid block containing the specimen to be sectioned. Then you place the solid block of the specimen embedded into the center of the block and put it on a sectional apparatus known as a microtome. You can think it as similar to a slicer used in the deli. Place the block in the arm of a microtome, and the arm will actually go up and down and pass the block over with a very sharp knife, sometimes a diamond knife, and you will slice the specimen into very thin section that can be placed on a microtome slide for light microscopy. Sometimes there are instruments called cryomicrotome. That means that the specimen under the block is actually frozen. Paraffin wax or the embedding media actually don't lose integrity due to the temperature. And then you put them onto a slide to do the next step. Fourth step in the sample preparation, there are a number of histological stains, dozens and dozens are available, all of which enhance the contrast of certain structure within the cell. I want to show you a few. Hematoxylin is very common. It's a nuclear stain and stains the nuclei of the cells a blue-violet color. It is a base, so it is attracted to acids like nucleic acid. So it binds, for example, to the DNA or RNA, which is an acidic molecule, and highlight the nuclei on the cell. The hematoxylin can also bind to some of the acidic amino acid like aspartic acid and glutamic acid. Another stain is eosin. It is an acid which binds to the basic macromolecules, and many cytoplasmic proteins are basic, so it colors the cytoplasm pinkish red. So you can easily see that as it is acidic, it binds the basic amino acid like lysine, arginine, very common amino acids present in many proteins. So it stains a pinkish reddish of the cytoplasm. These two stains are often used together and commonly known as H&E stain. And here see an example of a stained section of a gastric mucosal body. And these are actually parietal cells under the Brightfield microscopy. Peripheral blood smears are commonly stained with another stain which is known as a Wright Giemsa stain, which is a combination of eosin and methylene blue. And the acidic nuclei stain blue, and the cytoplasm is a pale pink. The red blood cells or erythrocyte which lack the nuclei stain a pale pink. These smears are used to identify and count white blood cells. So this is a very typical type of stain that is routinely used. The next one, another commonly used, is a benzidine, which is a stain that binds to heme-containing proteins. So you know that hemoglobin is a heme-containing protein. So you know the blood cells contain the heme. And the slide is a stage 35 xenopus embryo stained with a benzidine. And you can see that this is the ventral blood island stained very, very well with the stain right here. So all these blood cells are right here, and it's a classic heme-containing stain. So that actually conclude the part 2A of the section 4.2, which is just the Brightfield microscopy. And we will start with the fluorescence microscopy, which is also under the section 4.2 light microscopy in the next section, which will be the part 2B of section 4.2.